The 10 steps of glycolysis. What is glycolysis? Glycolysis is a metabolic pathway that converts glucose into pyruvate, releasing energy in the form of ATP and NADH. This process occurs in the cytoplasm of cells and does not require oxygen, making it both an aerobic and anaerobic process. It is the first step in cellular respiration, a universal process in living organisms. What are the 10 steps of glycolysis? Glycolysis is a 10-step process that breaks down glucose into pyruvate, producing ATP and NADH. Here's a summary of the steps. The process begins with phosphorylation of glucose. Glucose is phosphorylated to form glucose 6-phosphate, using ATP with the enzyme hexokinase. Isomerization of glucose 6-phosphate. Glucose 6-phosphate is converted into fructose 6-phosphate by phosphoglucose isomerase. Phosphorylation of fructose 6-phosphate. Fructose 6-phosphate is phosphorylated to form fructose 1,6-bisphosphate using ATP with the enzyme phosphofructokinase. Cleavage of fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is cleaved into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by aldolase. Isomerization of dihydroxyacetone phosphate. Dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate by triose phosphate isomerase. Oxidation of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is oxidized by glyceraldehyde 3-phosphate dehydrogenase, forming 1,3-bisphosphoglycerate. And NAD+ is reduced to NADH. Phosphorylation of 1,3-bisphosphoglycerate. The phosphate group on 1,3-bisphosphoglycerate is transferred to ADP, forming ATP and 3-phosphoglycerate, catalyzed by phosphoglycerate kinase. Isomerization of 3-phosphoglycerate. 3-phosphoglycerate is converted into 2-phosphoglycerate by phosphoglycerate mutase. Dehydration of 2-phosphoglycerate. 2-phosphoglycerate is dehydrated to form phosphoenol pyruvate by enolase. Decarboxylation of phosphoenol pyruvate. Phosphoenol pyruvate is decarboxylated to form pyruvate by pyruvate kinase, also producing ATP. What are the 10 enzymes of glycolysis? Glycolysis process involves several key enzymes that catalyze each step. The main enzymes are hexokinase, phosphoglucose isomerase, phosphofructokinase, aldolase, triose phosphate isomerase, glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and pyruvate kinase.